Good evening, class. I will be presenting on armodafinil, a drug that was discovered on accident, but became a first-line pharmacological therapy in treating excessive daytime sleepiness. So, excessive daytime sleepiness is found in narcolepsy, which affects about 1,000 to 2,000 people worldwide. Only narcolepsy type 1 is discussed here, so just a disclaimer. Narcolepsy type 1 is unique in that it includes cataplexy, where a strong emotion causes one's muscles to suddenly go limp. There is a gene which significantly increases one's chances of developing narcolepsy. And this gene is essential in the important for immune function. Thus, this is why scientists suspect that narcolepsy is caused by an autoimmune disorder. So, hypocretin is a neurotransmitter that is only produced under the hypothalamus and essential for regulating a proper sleep-wake cycle. Thus, it is no surprise that narcolepsy is caused by a loss of hypocretin-producing neurons. As depicted in this picture, we see a significant reduction in hypocretin-producing neurons in an individual with narcolepsy. The autoimmune theory states that the immune system is provoked in such a way that it falsely identifies hypocretin-producing neurons as foreign and destroys them. So, in 1974 France, two chemists were screening molecules in search of analgesics, and they identified a new molecule known as adrafinil. So, the earliest testing was done on mice. So forget about the whole drug discovery and optimization process, because back in the day, you went straight to in vivo testing. And they found that administering adrafinil increased locomotor effects, but they could not find evidence for stereotyped behavior or peripheral sympathomimetic effects, which were common in other stimulants. After this, kinetic studies were carried out, which identified modafinil as the active metabolite. After this, armodafinil was found to have a threefold higher affinity for the dopamine active transporter, and was suggested to be the longer lasting and more metabolically stable enantiomer, and thus the drug modafinil was born. So the two pharmacologists that discovered adrafinil suggested the mechanism of action might be an agonistic effect on the alpha-1 adrenergic receptor. To test this hypothesis, 3 alpha-1 adrenergic receptor antagonists were injected along with adrafinil. All 3 inhibitors prevented alpha-1 adrenergic hyperactivity, which suggests adrafinil is an agonist for the alpha-1 adrenergic receptor. Afterwards, however, multiple in vitro assays known to be responsive to alpha-1 adrenergic agonists found adrafinil to be inactive. So it appears that there is more to be understood about adrafinil than what meets the eye. Modafinil is also thought to interact with the dopamine active transporter, or DAT. In silico experiments reveal insight into the binding conformation of both modafinil enantiomers as depicted below. Both docked in the primary binding pocket of the DAT, and thus prevents the endogenous ligand dopamine from being reuptake into the presynaptic neuron. So because of this similarity in binding between the two enantiomers, it follows that the chiral sulfonyl group will point in a different direction. And the in silico results suggest that the oxygen is a hydrogen bond acceptor on armodafinil for the phenolic H on tyrosine 156. And this interaction is not seen in S-modafinil. So, studies were then carried out, in vitro studies, on COS7 cells expressing a mutant DAT, where tyrosine 156 was replaced with phenylalanine. The hypothesis was that if armodafinil is a hydrogen bond acceptor for tyrosine 156, then changing this to a phenylalanine should significantly decrease the affinity for armodafinil, whereas S-modafinil should show little change. And their results did find that S-modafinil affinity was cut in half, but more significantly, armodafinil affinity showed a 14-fold decrease. So, it was found that the wake-promoting effects of another DAT inhibitor stimulant known as amphetamine, but not those of modafinil, were antagonized by the dopamine receptor antagonist allopurinol. From this demonstration, amphetamine occupies the DAT, which increases extracellular dopamine concentration, allowing more dopamine to bind to the dopamine receptor and increase wakefulness. Introduce haloperidol, and it displaces dopamine, and thus the wakefulness is now antagonized. However, with armodafinil, when it's administered with haloperidol, it still promotes wakefulness. So it again, it seems that we have conflicting results here, and that indicates that there's more to be understood about armodafinil than we know now. So on to some pharmacokinetics. Armodafinil has 3 hydrogen bond acceptors and 1 hydrogen bond donor, and the same is seen in methylphenidate. Methylphenidate and amphetamine are older but still commonly prescribed stimulants for treating narcolepsy. And methylphenidate, although it has the same amount of hydrogen bond acceptors and donors as armodafinil, its topological polar surface area is about half of that as armodafinil. Amphetamine only has 1 hydrogen bond donor and 2 hydrogen bond acceptors, is if you count both H's, and it has significantly less topological polar surface area, as we would expect. So these drugs pass Weber's parameters and Lipinski's rules with flying colors, but we must remember that these are CNS drugs, and that they can't be pushing the parameters set forth in those rules, because they need to be very small and lipophilic if they are to permeate the blood-brain barrier. So many CNS drugs make use of amine functional groups, and this is seen in methylphenidate and amphetamine. You see that their topological polar surface area is very low, which should hinder their ability to be circulated throughout the body. But in fact, in vivo, in mainly a matrix of water, the amine can be ionized, and this greatly enhances the water solubility for these drugs, allowing them to move throughout the body very efficiently. And since the amine is ionized at physiological pH, there's a significant amount of molecule that is not ionized, and it can pass through the gut, the fatty cell membrane, and especially in the brain, it can pass through the blood-brain barrier very effectively. Now, this is not seen in armodafinil, as it is an amide, but it is more polar, so this should help its hydrophilicity in getting circulated around the body. However, it cannot go back and forth between hydrophilic and lipophilic because it only has one form, its neutral form in solution. And so thus synthesizing armodafinil analogs with amines may help its bioavailability in the brain. However, the amide group does participate in a pi stacking interaction with the phenyl group in the binding site. So this may cause armodafinil to have different pharmacodynamic interactions with the dopamine active transporter. So armodafinil is an example of a drug that did not proceed through the textbook drug discovery process. And so even though it was a failure as an analgesic, it is important, I guess these pharmacologists provided a great example of just having an open mind, and they were able to see that adrafinil could have a beneficial effect for something that we aren't testing for right now. And so they came back and they're like, okay, let's test this thing as a stimulant. And in turn, they created a first-line pharmacological therapy that has benefited narcoleptic individuals from around the world. And as indicated by the alpha-1 adrenergic interactions and interactions with the dopamine active transporter, we have challenging pharmacodynamic findings. And so there is a great amount to be understood about armodafinil and how it interacts in our body. So in the future, armodafinil may be administered via nanocrystal hydrogels to increase its bioavailability. Furthermore, other therapeutics in treating narcolepsy may also be administered intranasally, such as hypocretin 1. Now, this may surpass armodafinil as a first-line pharmacological therapeutic because it treats the problem closer to the source, as it is replacing the neuron that is no longer able to be produced by individuals with narcolepsy. And this new therapy has delivered results. So this may be a promising new therapy in treating narcolepsy. And here are my references. I hope you enjoyed this presentation.